Hello and welcome to Biology 8. Now this is a recap episode, and it's also an episode where I'm going to go through the functions of all the organelles that we've covered so far. I've got a lot of pictures, I've got some electron microscope images, and I'm going to go into considerably more detail than I've gone through before. The structure will be that I go through the text section first, then I'll show what images I have, if any, for that particular organelle, and then we'll go through the text section again for the next organelle. So that's going to be the structure of this episode. So the first organelle we're going to consider is the plasma membrane. And the plasma membrane is found on the surface of animal cells and just inside the cell wall of plant cells and prokaryotic cells. It's made mainly of lipids and protein. Lipid bilayers form through the process of self-assembly. The cell membrane consists primarily of a thin layer of phospholipids which spontaneously arrange so that the hydrophobic tail regions are isolated from the surrounding polar fluid, causing the hydrophilic head regions to associate with the intracellular cytosolic and extracellular faces of the resulting bilayer. This forms a continuous spherical lipid bilayer. Lipid bilayers are impermeable to polar molecules. The arrangement of hydrophilic heads and hydrophobic tails of the lipid bilayer prevent polar solutes, e.g. amino acids, nucleic acids, carbohydrates, proteins and ions, from diffusing across the membrane, but allows for the passive diffusion of hydrophobic molecules. This affords the cell the ability to control the movement of these substances via transmembrane protein complexes such as channels, pores and gates. Regulates the movement of substances into and out of the cell, and it also has receptor molecules on it which allow it to respond to chemicals like hormones. So for each of the organelles, I've done this little bit in capital letters as well, just to try to choose what I thought was the most important thing. So it's got a lipid bilayer. So let's have a look. See, we've got some pictures. Here's my picture. First of all, just at the beginning, I've actually put all my pictures that I've done so far into this as well. So so it's the cell surface membrane just in the purple there, and so I won't dwell on it too much. Just going through it anyway, and then on the plant cell as well. You can see it's two layers there instead, because obviously the plant cell's got that very strong cell wall as well. Okay, so like I said, we talk about the plasma membrane. So it's this membrane here with the cytoplasm inside the cell. And here's an actual diagram, which kind of well, my diagrams are still diagrams, but it's obviously slightly different level. But we can see these are the tails here of these things, the hydrophobic tails and the hydrophilic heads. So it's a combination of things that love water and hate water. So that's really the key thing here, and it's to keep the integrity of the cell. This is the bilayer in detail, and I think I've actually got another one as well. Yeah, this was what I showed in episode 4. Similar kind of picture. You can see there's proteins there on the cell membrane, and you can also actually see these little heads and the tails in the membrane, which is very, very cool. And I'm pretty sure that is the last one. Yes, we're going to be on the cell wall next. And the advantage is that I won't actually spoil pictures, because it will just be my pictures, which obviously next one, the cell wall. Okay, so it's got considerable tensile strength and is made of mainly the carbohydrate cellulose. Up to 3 layers of strata may be found in plant cell walls. The outermost layer is the middle lamella, a layer rich in pectins, so gelling agent. This outermost layer forms an interface between adjacent plant cells and glues them together. The second layer is the primary cell wall, generally a thin, flexible and extensible layer formed while the cell is growing. The secondary cell wall, which is a thick layer formed inside the primary cell wall after the cell is fully grown. So one of the things I thought of when I read that was like the primary cell wall seemed like your sort of first set of teeth, and the secondary cell wall seemed like a second set of teeth. That was my initial response to it. Okay, so it is not found in all cell types, the secondary cell wall. Some cells, such as the conducting cells in xylem, possess a secondary wall containing lignin. I'm going to go for lignin, which strengthens and waterproofs the wall. Okay, so let's see what we've got in the picture front for cell wall. So here we go. We can see we've got the cell wall there, surface membrane, and the cytoplasm. So just another layer. So it's a bit like, yeah. Okay, here's the picture of what we were speaking about. You can see the pectin there. You can see it's made of cellulose in those microfibers. And yeah, and I'm pretty sure that's the only one I've got. Yep. Okay, so there's the nucleus. So let's talk about the nucleus. Okay. So the nucleus is a large organelle surrounded by a nuclear envelope. It's a double membrane, which contains many pores. The nucleus contains chromatin and often a structure called the nucleolus. Chromatin is made from proteins and DNA. DNA controls the cell's activities. The pores allow substances, e.g. RNA, to move between the nucleus and the cytoplasm. The nucleolus makes ribosomes. The nucleus is a membrane-enclosed organelle found in eukaryotic cells. It contains most of the cell's genetic material, organized as multiple long linear DNA molecules in complex arrangements with a large variety of proteins, such as histones, to form chromosomes. The nucleus is the control center of the cell. The main structures making up the nucleus are the nuclear envelope, a double membrane that encloses the entire organelle and isolates its contents from the cellular cytoplasm, and the nuclear skeleton, which includes nuclear lamina, a network within the nucleus that adds mechanical support, much like the cytoskeleton, which supports the cell as a whole. Because the nuclear membrane is impermeable to large molecules, nuclear pores are required that regulate nuclear transport of molecules across the envelope. The nuclear lamina regulates important cellular events such as DNA replication and cell division. Additionally, it participates in chromatin organization, and it anchors the nuclear pore complexes embedded in the nuclear envelope. The nuclear lamina is associated with the inner face of the bilayer nuclear envelope, whereas the outer face stays continuous with the endoplasmic reticulum. Similar to the cytoplasm of a cell, the nucleus contains nucleoplasm, nucleus sap, or karyoplasm. The nucleoplasm is one of the types of protoplasm, and it is enveloped by the nuclear membrane of the nuclear envelope. The nucleoplasm is a highly viscous liquid that surrounds the chromosomes and nucleoli. Many substances, such as nucleotides, necessary for purposes such as the replication of DNA, and enzymes, which direct activities that take place in the nucleus, are dissolved in the nuclear plasm. The soluble liquid portion of the nuclear plasm is called the nuclear hyaloplasm. Okay, so we're going to go back, and here's my nucleus, and you can see the pore there, the chromatin, the nucleolus there, and the envelope. Let's move on to the this one, and so we can see there we've got the chromatin. There seems to be different types of chromatin, heterochromatin and euchromatin. We've got the ribosomes there, the outside, the outer membrane, and the inner membrane, nucleolus. Large structure there, and with the nucleus's version of the cytoplasm, the nucleoplasm. Do we have any more on that? Oh yeah, we've got this one as well. So again, looking at the nucleus, we've got the nucleolus there, nuclear pores, chromatin, same sort of stuff. And then we're going to be on to the lysosome. So let's talk about that. So the lysosome. Lysosome is a round organelle in animal cells surrounded by a membrane with no clear internal structure. Contains digestive enzymes that are kept separate from the cytoplasm by the surrounding membrane and can be used to digest invading cells or to break down worn-out components of the cell. Lysosomes can be described as the stomach of the cell. They are cellular organelles that contain acid hydrolase enzymes that break down waste materials and cellular debris. Okay, so we can see no clear internal structure. It was difficult to find images for the lysosome. So let's now talk about the ribosome. Ribosome, a very small organelle that either floats free in the cytoplasm, attached to the RER, or inside the mitochondrial matrix. It is the site where proteins are made. It is a large and complex molecular machine found in all living cells that serves as the primary site of all biological protein synthesis, translation. Ribosomes link amino acids together in the order specified by messenger RNA, mRNA, molecules. Ribosomes consist of two major subunits. The small ribosomal subunit reads the mRNA, while the large subunit joins amino acids to form a polypeptide chain. Each subunit is composed of one or more ribosomal RNA, that's rRNA molecules, and a variety of proteins. The ribosomes and associated molecules are also known as the translational apparatus. Okay, so let's see. We've got a ribosome. Not too much, I don't think. Nope. Okay, so now we're going to move to the rough endoplasmic reticulum, I believe. Yes. Okay. Okay, and I've done this with both of them here. So the rough endoplasmic reticulum, a system of membranes enclosing a fluid-filled space. The surface is covered with ribosomes. It folds and processes proteins that have been made at the ribosomes. So you can see the ribosomes there on the edge there. And yeah, and so this is a micrograph of the endoplasmic reticulum network around the nucleus. So we've got the dark circles being mitochondria. You can see these nice, it almost looks like, it looks exactly like a fingerprint. That's what it looks like to me. But of course it's actually on a lung, a human lung, I believe. There we go, the rough endoplasmic reticulum network. Let's see what else we've got here. Okay. Smooth. And I did put a little more in here actually. So let's see. There we go. Now the smooth endoplasmic reticulum, the SER, similar to the RER but with no ribosomes, synthesizes and processes lipids. So the outer nuclear membrane is contiguous with the endoplasmic reticulum. While this is physically linked, the outer nuclear membrane contains proteins found in far higher concentration than the endoplasmic reticulum. Endoplasmic reticulum occurs in most types of eukaryotic cell, but it's absent from red blood cells and spermatozoa. The outer cytosolic face of the RER is studded with ribosomes that are the sites of protein synthesis. RER is especially prominent in cells where active protein synthesis occurs. The SER lacks ribosomes and functions in lipid and carbohydrate metabolism. Okay, so no ribosomes there in the smooth endoplasmic reticulum. Okay, so now we move on to the vesicle. Vesicle, a small fluid-filled sac in the cytoplasm surrounded by a membrane. It transports substances into and out of the cell via the plasma membrane and between organelles. Some are formed by the Golgi apparatus or the endoplasmic reticulum, whilst others are formed at the cell surface. For example, the secretory pathway is a series of steps a cell uses to move proteins out of the cell, a process known as secretion. The path of a protein destined for secretion has its origins in the RER, a membrane-bound compartment in the cell. The protein then proceeds through the many compartments of the Golgi apparatus and finally ends up in a vesicle, then transiently fuses at the cell plasma membrane via permanent plasma membrane structures called porosomes, depositing the proteins outside of the cell. And we'll move back to our pictures here. So there's the membrane and there's the vesicle there, the plasma membrane of the cell. And I actually did find this vesicle here in Coleus Blue May. This is a vesicle, and it's a mint. I believe this is mint, as in the plant. So you can see, can't see too much, but you can see those little sort of spherical objects there in that close-up there. Okay, so I think that's the only one. Yep. So Golgi apparatus time. So the Golgi apparatus, a group of fluid-filled fattened sacs. Vesicles are often seen at the edges of the sacs. It processes and packages new lipids and proteins, and also makes lysosomes in animal cells. In general, cells synthesize a large number of different macromolecules. The Golgi apparatus is integral in modifying, sorting, and packaging these macromolecules for cell secretion, that's exocytosis, or use within the cell. It primarily modifies proteins delivered from the RER, but is also involved in the transport of lipids around the cell and the creation of lysosomes. It packages and labels items which it then sends to different parts of the cell. So let's see what we've got. So off to the edge of the vesicle, like I've mentioned, and we can see it's got some really good pictures, again from this mint plant I found. You can see the structure of the Golgi quite clearly, and let's see what else we've got. Here's the Golgi in the human leukocyte. You can see they're quite fat structures. Okay. So now we move on to mitochondria. Okay. usually oval-shaped with a double membrane. The inner membrane is folded to form structures called cristae. Inside is the matrix, which contains enzymes involved in respiration. There are 5 distinct parts. The first is the outer mitochondrial membrane. The second, the intermembrane space, which is the space between the outer and inner membranes. The third is the inner mitochondrial membrane. The fourth, the cristae space, formed by foldings of the inner membrane. And finally, the fifth is the matrix, space between the inner membrane. Although most of the cell's DNA is contained in the cell's nucleus, the mitochondrion has its own independent genome. Also contains the ribosomes in the matrix. Okay. So the inner membrane contains proteins with 5 types of functions. The first is those that perform the redox reactions of oxidative phosphorylation. The second is adenosine triphosphate synthase, which generates ATP in the matrix. The third is specific transport proteins that regulate metabolite passage into and out of the matrix. The fourth is protein import machinery. The fifth is mitochondria fusion and fission protein. Its function is as a site of aerobic respiration, where ATP is produced. Mitochondria are found in large numbers in cells that are very active and require a lot of energy. So there we can see, this is a picture that I created. We've got the outer membrane, inner membrane, the cristae, and matrix in the middle. And we've got several, and we can see some, this lung picture, some mitochondria. You can see these structures very, very clearly. That's a good one. Really close up, you can see all the structures of the mitochondria. That's the lung. And here's another picture showing the inner membrane, the outer membrane, the cristae, and the matrix, which is in the core. Okay. Well, that's not. Okay. So a chloroplast now is the next organelle to look at. Chloroplast, a small flattened structure found in plant cells. It's surrounded by a double membrane and has membranes inside called thylakoid membranes. These membranes are stacked up in some parts of the chloroplast to form grana. Grana are linked together by lamellae, thin, flat pieces of thylakoid membrane. It is the site where photosynthesis takes place. Some parts of photosynthesis happen in the grana, and other parts happen in the stroma, a thick fluid found in chloroplasts. In photosynthesis, the photosynthetic pigment chlorophyll captures the energy from sunlight and stores it in the energy storage molecules ATP and NADPH. They also perform fatty acid synthesis, amino acid synthesis, and the immune response in plants. They're a type of plastid. Okay, let's look at the chloroplast now. So here's my drawing here. I've got the stroma, lamellae, granum, grana. Okay, and chloroplast and mitochondrion are a key in endosymbiotic theory. Here's a very good drawing of it. We can see the granum, similar to a stack of coins. The outer membrane, inner membrane space, inner membrane, and the stroma of the fluid there. And yeah. So a stack of thylakoids. Okay. And here's a slightly more simplified diagram as well. Okay. So now we move on to the centriole. Small hollow cylinders that contain a ring of microtubules, tiny protein cylinders. An associated pair of centrioles surrounded by an amorphous mass of dense material makes up a compound structure called a centrosome in animal cells. Remember, of course, lysosomes are not in plant cells. There's this, they do have a vacuole there. Okay, anyway. The position of the centriole determines the position of the nucleus and plays a crucial role in the spatial arrangement of the cell. The last common ancestor of all eukaryotes was a ciliated cell with centrioles. Okay, and here my diagram, unfortunately it was incorrect. So there's only actually 9 of these here. And here's a centriole from the brain of a mouse. And we can see we've got 9 there. 9 doublets. Okay. Okay, so cilium now. Cilia are small hair-like structures found on the surface membrane of some animal cells. They have an outer membrane. Inside cilia and flagella is a microtubule-based cytoskeleton called the axoneme. There are 2 types of cilia. Primary cilia has a ring of 9 outer microtubule doublets called a 9+0 axoneme, and the axoneme of a motile cilium has 2 central microtubule doublets in addition to the 9 outer doublets called a 9+2 axoneme. So in motile cilia, the microtubules allow the cilia to move. This movement is used by the cell to move substances along the cell surface. Eukaryotic cilia are structurally identical to eukaryotic flagella, although there are distinctions with regard to function or length. So we can do it. There's a side and there's the cross section. Hair-like structures, 9 pairs of protein microtubules. Yeah. And we may have some images as well. Here's some images from mammal cell. And we can see the 9+2 structure here. So finally we have flagellum. Flagella on eukaryotic cells are like cilia but longer. They stick out from the cell surface and are surrounded by the plasma membrane. Microtubules contract to allow flagella to move. There are 3 types of flagella: bacterial, archaeal, and eukaryotic. Flagella are organelles defined by function rather than structure. Their primary role is locomotion, but they also have a function as a sensory organelle. Okay, and this was my picture. So yeah, anyway, so that's going to be the end of Biology 8. So thanks for watching.